Now we will focus on cellular organelles, subtopic 1.2, and we'll be using a typical animal cell to do so. Now, what exactly do I mean by a typical animal cell? What do you mean by a typical animal cell? Simply speaking, in the scientific community, whenever we talk about something as being typical, we think of it as general. Now, when I draw the structures in, here's the deal. Not every animal cell is going to contain all of these structures. But these are all the structures that are going to be found commonly in animal cells. Now, if that did not make sense, perfectly fine. That does not have to make sense. Now, the best way that I have found to go through cellular organelles of a typical animal cell in the most amount of detail that you need for this specific course is to first draw a diagram of a typical animal cell and explain to you where exactly each and every organelle is located, and then we're going to be diving into their functions in a lot more detail. Right, let me start off by drawing a huge, almost oval type of shape. Now, animal cells, they're not regular in shape, but that's a little too irregular. So, smoothen things out. Okay, that's better. Okay. It's not a perfect circle, it's not a perfect oval, it's a general type of cell. Now, I'm going to start off with the main part, which is going to be the nucleus. For better understanding, I'm going to zoom in to the cell. Don't worry, I'll zoom out in a bit. And here we have the nucleus. I'm drawing this like this due to a reason. That reason I'll come back to very soon. Boom. And there's something like this in the middle. And then within the nucleus, you have these things. I'm going to use that color and I'm going to use this color on top for those. And then this nucleus is actually a double membraned structure, meaning it has two membranes. That's our first membrane, and then here we have our second membrane. Oops, sorry, that turned to a line. And then from this second membrane, something interesting is born. Something that looks like this is born. Like so. Let me draw that in. I will try to do the maximum justice I can with this. And I'm not going to draw it all the way around. I will draw that and maybe a little bit here. I'll name this, don't worry. So something like this and like this. So all you have to know from this is that the nucleus is actually a double membrane structure, and there's something like this in the middle. I'll name that as well. And as for the other organelles, we'll get to them afterwards. So first of all, let's start by naming these things that are present in front of us. What color should I use? Use a pretty pink then? Okay, that's fine. Right. Starting off with the cellular membrane, also referred to as the plasma membrane at this level. So you have to be aware of that as well. This can also be called the plasma membrane. They mean literally the same thing, there's no difference. Plasma membrane. And now let's label these things. So this middle ball that you see is actually known as the subnucleus. A much better word for it was always nucleolus. Nucleolus. And this is your entire nucleus. That's your nucleus. So the nucleus is simply comprised of the nucleolus and chromatine. And chromatine. Now, what exactly are these structures that branch off of the second envelope of the nucleus? So a very important distinction that I'm going to make, and a very important that you have to put thing that you have to put into your mind, is the nucleus is not a single membrane structure, and as for the cell membrane, that is also not a single membrane structure. That part we'll get to in a bit. I know I just drew one layer here, but in reality there's a second layer as well. We'll get to that when we discuss the organelles in detail. But here distinctively I have drawn the nucleus as being a double membrane structure. And have you noticed this? I left some spaces here, and you can see that things can move in and out of these spaces like so. Typically a space like this is known as a nuclear pore. Is known as a nuclear pore. Simply a pore. There's a specific reason why we need pores and why things need to travel back and forth from the nucleus like so. We'll get to those things in protein synthesis and stuff. But for now, all you have to know is we have a nuclear pore. Now, back to this. I keep distracting you and myself from this. Let me pick a nice color. Okay, that works. Right. So here from the second membrane, something known as the endoplasmic reticulum is born. Endoplasmic reticulum, or ER for short. Endoplasmic reticulum, ER. Now we actually have two types of endoplasmic reticuli. Two types of endoplasmic reticulums. Here, I'm going to leave this ER as is. I'm not going to do anything to this. Not going to do anything to this. But this ER and this ER, I'm going to do something like this. I'm going to place small dots on them like so. Small dots on them like so. So on the reticulum. And I would really recommend, I would really advise going on Google and giving a small search phrase, endoplasmic reticulum 3D, so that you can actually get an idea of what they look like. This is just a 2D image. No, just give it a go and you'll get a better perspective of all of this. So now I made this like this. I'm going to actually remove these three things. Let's get rid of those things. Right. Now do you see, this is no longer like this. This guy looks smooth. This guy looks smooth. But here now this thing has a rough appearance. Now these things that I actually put in here are ribosomes. We'll be talking about ribosomes in more detail as well. These things that I put here are known as ribosomes. Whenever ribosomes are impregnated onto the endoplasmic reticulum, we call that the rough endoplasmic reticulum. We call that the rough endoplasmic reticulum, or, if you have guessed it, RER for short. So I'm going to get rid of this and I'm going to place in an R here. So rough endoplasmic reticulum. In contrast to that, this guy, what about that guy? He's not rough. He didn't look rough. I'm going to move nucleolus down a bit. And I'm going to get rid of that. No, here we have still, it's an endoplasmic reticulum. Nothing has changed, but typically we refer to this as being the smooth endoplasmic reticulum. Smooth ER. What's the main difference? This rough endoplasmic reticulum gets its name and form and shape from these things. What are those things? Those are ribosomes. Do you see when ribosomes are impregnated onto this, like so, what's happening? Simply speaking, it has a rough appearance. So for further clarity, ribosomes, yes. Ribosomes, no. That's the difference. If you have ribosomes, it's rough. It has a rough appearance. RER, rough endoplasmic reticulum. I'll foreshadow a little bit. The RER contains ribosomes, therefore I'm pretty sure it's responsible for protein synthesis. Smooth ER has no ribosome, so there's no protein synthesis happening in the smooth ER, SER. If those statements made no sense, please bear with me. We'll get to those things. Right. Now, those are the main things about these reticuli and the nuclei that are in a plant cell. Now we're moving on to a set of different organelles. The first different organelle I'm going to talk about is something that looks a little bit like the ER. Now, in fact, this ER can break like so and produce these vesicles. Vesicles have many different functions inside cells. We'll talk about that as well. But from those vesicles, basically, when things come broken down, you get certain things that look like this. It's almost like a Wi-Fi signal. Like so. This Wi-Fi looking thing is known as your Golgi apparatus. Golgi, or some people call it Golgi. Golgi apparatus, or you can call it Golgi bodies. Golgi bodies. And you can see these small little things. This entire organelle as a whole is known as the Golgi body. And here you can see a little small Golgi vesicle. I'm going to write that in small handwriting. Golgi vesicle. It's a small vesicle. That's all. This specific organelle has a very important function. We'll talk about that when we talk about this organelle in a little bit more detail. And something very interesting that we're almost going to miss every single time is this. The cytoplasm. I don't know why I wrote it in a corner. Let me get it here. Okay, I'll take it here. And here, obviously, we have the cytoplasm. That's your cytoplasm. Any other things? What other organelles can a plant cell, an animal cell possibly contain? So now we talked about ribosomes that were bound to this. No, apart from that, we can have ribosomes just on the cytoplasm. We typically refer to those things as being free ribosomes. As being free ribosomes. Typically free ribosomes. And I'm going to draw something very interesting. And it looks like this. Now you might not think it's that important at this level, but it is more than important. So that looks like what? If that looks like a mitochondrion each, congratulations, you have figured it out. Here we have mitochondria responsible for cellular respiration, responsible for the production of energy in the form of ATP. Apart from ribosomes, we also have another type of vesicles. These are known as, why is it not so thick? Right. These are known as lysosomes. And I'm also going to introduce a rather new set of organelles. Now this you have actually not seen before. Closer to the middle part of the cell, we have things that look like cylindrical structures. Don't think of this too much. I'll go into more detail when it comes to this. But for now, all you have to know is these are known as, I don't have space. Okay, place it here. These organelles are actually known as centrioles. That's typically two centrioles arranged in a 90-degree format. Do you see? One is facing this way, the other is this way, and between them there's a 90-degree angle. That's typically how they're placed. Why? We'll talk about that later. So I'm just going to mention that centrioles are placed in a 90-degree format like so. Now sometimes this, what do you call it? Cell membrane can become like this. Now do you see? It's ready to produce this thing. What do you call that thing? That thing is known as a secretory vesicle. A secretory vesicle. Secretory vesicle. Simply a secretory. What exactly is it secreting? That's not important. Secretory vesicles can branch off from Golgi like this, from the endoplasmic reticulum itself, and so forth. And they can also just be produced from the cellular membrane itself. It's not that much of a big deal. And with that, these are the main organelles. I'm going to mention that again. These are the main organelles that you have to focus on for advanced subsidiary levels. And believe me, yes, there's a lot more organelles when it comes to the cell, an animal cell, but these are the main things that you have to focus on. And now the next step would be to go over each and every one of them in a lot more detail. Starting off with the cell membrane. I'm gonna set this to 110, and yes, so that's your overall picture. And now we'll be focusing on each and every one separately. So first of all, we have the cellular membrane. Okay, I'm going to improve that a little bit. Let's move membrane all the way up until here, and I'm going to add the term surface in between. Now the cell surface membrane is actually 7 nanometers in width. It's not that large at all, as you can see. How exactly would I describe the cell membrane? I will describe the cell membrane like this. So if there was a 2-mark question asking you to define what is meant by the cell surface membrane or describe the cell surface membrane, this is exactly how you would write your answer. The most important thing is that you have to mention that the cell membrane, oh, did I write partial membrane? My God. Mine is numb. Sorry. A partially permeable membrane. Partially permeable membrane. It's only semi-permeable. A partially permeable membrane that regulates the exchange of materials. What do I mean by materials? Any form of biological molecule, and that can also include ions, between the cell and its surroundings. Now you know the only thing that a cell has to do is simply do metabolic functions, and for that cell to actually do metabolic functions, it has to communicate with its environment. So it's going to be between the cell and its surroundings. It has to take things in from the surrounding to do metabolic processes. Whatever waste products the cell has actually produced, it has to release into the environment. There's a lot of communication between the cell and the surrounding. Right. Now to a point that I made previously. Remember how I specifically drew the nucleus as being a double membrane structure like so? It was not a problem at all. Hence, I will now show you why I talked about the cell membrane, cell surface membrane, also as being a double membrane structure. So here's the deal. It is also a double membrane that looks like this. So I'm going to zoom in to these things, and I'm going to draw this double membrane in an interesting manner, possibly in a way that you have not seen this before. So we have circles. Circle 1, circle 2, circle 3, circle 4, 5, 6, 7, 8. And all of these circles have two tails. Tail, 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 tail. And it's a double membrane, remember? So we have that kind of cellular structure. To distinguish that a little bit better, I'm going to move it like this. Okay. Now, do you see? This is the first membrane. That is the second membrane. Now, these little molecules that we have here are known as phospholipids. Phospholipids. Okay. And phospholipids have a very distinguished reputation. Now, you see, this is a single phospholipid. You'll learn about phospholipids in detail under biological molecules, which is chapter 2, spread 2 of this course. But for now, all you have to know is, you see, there's a head, and we typically call this a tail. There's a head and two tails, a tail or two tails in any form of way. So we typically call this a hydrophilic head. Hydrophilic. When you hear that term, what do you think of? What do you mean by hydro? Hydro means water. Philic means to love. Does it though? Does philic mean to love? How exactly are we going to know that philic means to love? It'll become obvious when I give you this. Now this is what you know as a hydrophobic tail. Hydrophobic tail. Phobia. Being scared of, not liking. So simply speaking, the tails are hydrophobic. They don't like water. The heads are hydrophilic. They do like water. That's the difference. Hydrophilic heads like water. Hydrophobic tails do not like water. So now these things are made out of phospholipids, and you can see we have two layers of phospholipids. No? So we typically refer to them as being a phospholipid bilayer. Not a monolayer, a bilayer consisting of two things. We have a phospholipid bilayer. That's the thing. Simply speaking, we'll touch up on the functions. Why do I always have thicker pens? Okay, that's better. So let's talk about the functions of a cell surface membrane. Now these are the exact same functions that you have known since your small days. Not exactly your small days, but previous grades. There's not that much of a difference to all of these things. So the first function would be the exchange of materials. So those would be molecules and ions. And this is a new thing, cell signaling. Now, when we talk about the cell membrane in a lot more detail, yes, we will. Now, this is the very basic idea of the cellular membrane that I just gave to you. It looks a little bit like this, but there's a lot more to this. There's a lot more to the cellular membrane. So once we get to that, you'll learn about glycoproteins that can be switched to this, other proteins that can be there on this bilayer. We call them transmembrane proteins. There's a lot happening, and those things promote a process known as cell signaling. Very simply put, cell signaling is where cells talk to each other. They talk to each other. That's the idea. So there's something known as cell signaling, and that is one of the functions of the cellular surface membrane, as well as during this talking process, they can recognize each other as well. So cell recognition, and there's something interesting. The cell surface membrane can form hydrogen bonds with water for stability. Hydrogen bonding. Moving on to the second organelle I would like to talk about, it's going to be, of course, the nucleus. The nucleus. I will give you a simple size definition. The nucleus is 6 micrometers in width. That's the width of the nucleus. Now, I'm not really going to draw the nucleus again. This is basically what the nucleus looks like. So I'm just going to source that out. Nucleus, 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 come on. This is what the nucleus looks like. Isolated, that's what it looks like. Let's start off with the key points. First of all, this will be the largest organelle that you're going to find. That's why it's 6 micrometers, not 6 nanometers. In comparison, remember the cell surface membrane is very thin, 7 nanometers only. But here the nucleus is going to be 6 micrometers. The nucleus is going to be 6 micrometers. Here, this is simply the largest organelle. The nucleus is surrounded by the nuclear envelope. Surrounded by the nuclear envelope. Now, these are the 2 layers I was talking about. 2 layers. And then this second layer is continuous with the RER, rough endoplasmic reticulum. I'm going to switch colors. Let's go pink. And remember we talked about nuclear pores. Nuclear pores, they're simply openings in the nucleus. Why do we need them? Should I explain now? Yes, I will. Has nuclear pores. Why exactly do we need pores? Because they facilitate mRNA and proteins leaving the nucleus. Leaving and other things entering as well, but it's mostly leaving in the cellular processes. mRNA proteins facilitate mRNA and proteins leaving the nucleus. Switch to a different color and I'm going to give you the final point. What do we have inside the nucleus? We have something known as the nucleolus. The nucleolus is only 0.5 micrometers in size. Obviously much smaller than the nucleus. The nucleolus functions in the synthesis of ribosomes. In the synthesis of ribosomes. So a very cool way of saying this. So I recently discovered this only recently. A very cool way of saying this would be facilitates. I'll write that better handwriting as well. Facilitates ribosomal biogenesis. My God. What does it mean? The exact same thing as this, but it's a much cooler way of saying it. So that's all about the nucleus. Continuing on with the nucleus, the other organelles that are going to be linked to it, like we talked about, is first of all going to be, no, that's 1, 2, that has to be 3, 2, 3, has to be the endoplasmic reticuli. So I'm simply, okay, let's call them the endoplasmic reticuli. ER commonly. Under ER commonly, we'll talk about the rough endoplasmic reticulum, RER. Rough endoplasmic reticulum. Okay, really apologize for the handwriting, but I know you can figure it out. Okay, points to this. I'm not going to get a second diagram, it's simply this, so I'll tell you what, I'll get a much smaller diagram. Whoops, what happened? I'm gonna get a smaller diagram. Okay, I'll just leave it here just for a better idea. So here we have the endoplasmic reticuli, the rough one first, and let me pick a nice color. We'll talk about the rough endoplasmic reticulum in a lot more detail. The first point that you can obviously see, and I mentioned, is that it is continuous with the second layer of nuclear membrane, and it has the rough appearance because ATS ribosomes are embedded. What on earth do you mean by ATS ribosomes? We'll talk about that as well, but for now all you have to know is it has ribosomes embedded to it, and that is why it has this rough appearance. That's why it has this rough appearance. And this is the most important thing. This is actually the site for protein synthesis. This is actually the site for protein synthesis. Why? Because ribosomes are the main protein producing factories, and the rough endoplasmic reticulum is full of them. The rough endoplasmic reticulum is full of them, and that is why we consider this as being the main site for protein synthesis. Now, the proteins synthesized here, they can actually be used outside of the cell or in the cellular membrane. So typically speaking, whatever protein that is produced in this, so in the endoplasmic reticulum, is packaged up into a vesicle, and this vesicle becomes a secretory vesicle, and this thing is actually going to be secreted outside. Most proteins produced in the RER are extrinsic proteins. That's the idea I'm trying to give you. Extrinsic proteins. Proteins utilized outside. Proteins utilized outside the cell. Proteins utilized outside the cell. Now, what about intrinsic proteins? The cell itself also needs proteins, so what about them? Those things are produced simply by these free ribosomes that you find inside, like so. Free ribosomes that you find inside, like so. Okay. What else? Ah, exactly what I said. I didn't write that as a point, no. Packages proteins into vesicles. Packages proteins into vesicles for transport. What about the second part of this, which is going to be the smooth endoplasmic reticulum? Handwriting is going to go haywire. I'm not putting that much effort into handwriting when it comes to this specific thing because it's information and information and information. Of course, if there are diagrams, I want to be more specific on that. No worries on that end. What exactly does the smooth endoplasmic reticulum do? This is the site for protein synthesis. The SER is simply the site 4. If that was proteins, this is going to be lipids. Site 4, lipid synthesis. Side 4, lipid synthesis. And we are also producing steroidal hormones. And obviously no ribosomes embedded, hence the smooth appearance. Smooth appearance. Along the same chain, we also talked about the Golgi apparatus. We'll talk about that as well. So line it here. It's number 4. Golgi apparatus. Golgi bodies, Golgi apparatus, same notion. Okay, I'm gonna draw it separately here. So they typically look like this. This is the exact same thing I have drawn in the diagram as well. No difference. A little bit closer to the edge. That's the only difference. I can fix that. Right. Golgi apparatus, they typically look like this. It's like an inverted Wi-Fi signal or a Wi-Fi signal. Not that much of a problem, not that much of a difference. Okay. The Golgi apparatus are simply a stack of flattened a stack oh my god. Stack of flattened stacks. Interesting. These sacks we actually call as cisternae. Not to be confused with cristae, which you will find in the future in mitochondria. Just giving you a warning. They are known as cisternae. Now here's the funny thing about the Golgi apparatus. This organelle is formed from another organelle. This organelle is simply formed from the intervention of another organelle, and that is the endoplasmic reticulum. Here's the deal. It is actually formed by vesicles that bud off from the endoplasmic reticulum, from the ER. What exactly is the function of this Golgi apparatus? This is an organelle that we have not really heard of before, not really put into consideration before. So why is this so important? Now here's the function of the Golgi apparatus. Modification of proteins. Processes other biomolecules. And it is capable of engulfing them in Golgi vesicles. Golgi or Golgi vesicles, same deal. I just like how Golgi sounds in my mouth, I guess. Most of the people I've also met do call it Golgi. So forgive me if Golgi seems weird. Golgi vesicles for transport to other parts of the cell. Or once it's in a vesicle, it can go outside the cell extracellularly as well. No issues. And also the Golgi apparatus is responsible for the production of lysosomes, glycoproteins. Now we talked about how it modifies proteins. When proteins get a glucose part added to it, we typically call them glycoproteins. And also glycolipids. Don't worry, you'll learn about glycoproteins, glycolipids, cellular membrane structures, and all that in a lot more detail. For now, this is just brushing off. And also, the Golgi apparatus is mainly responsible synthesizes lipids. Smooth endoplasmic reticulum is also responsible for synthesis of lipids and steroidal hormones, as well as the Golgi apparatus as well. Moving on to now that chain of organelles is over, starting from the nucleus to the cell membrane. Now, do you see they all carried similarities? We had the cellular membrane, then the nuclear membrane, two membranes, glioles, Golgi apparatus. All of them worked in combination to each other. Of course, all organelles do work in combination to each other. They can't really function alone. That's not my point. What I'm trying to say is they followed a common theme. Like so. That theme ends here with the discussion of mitochondria. A mitochondrion is also pretty large, actually. It's 1 micrometer in length. Now I'm going to draw a mitochondrion in a lot more detail than I did before. And it's very easy to draw a pretty nice looking mitochondrion as well. You simply start off with an oval, and then you have to just follow that oval up, and then with these specific intermediate parts, you just have to go through like this. See? Pretty nice. Now I'm going to label the parts. Remember how I told you that we have infoldings? These infoldings are known as cristae. Not cisternae, but cristae. Okay. Here we have the outer membrane OM. Then we have the inner membrane IM. And this cytoplasm type of thing is actually known as the mitochondrial matrix. And in between the outer membrane and the inner membrane, what do we have? We have the intermembrane space. Pretty simple. Outer membrane and inner membrane. In between that, we have the intermembrane space. IM space, intermembranal space. And inside, in the middle, we have the matrix. In the matrix, we have all the stuff. Mitochondrial ribosomes, mitochondrial circular DNA, other proteins, other solutions. Everything is suspended in this. Now, what is so special about the mitochondria? I'll use star points here. This is the site for cellular aerobic respiration. That's the main function of mitochondria. And we'll talk about the mitochondrial matrix. Has a matrix containing 70s ribosomes. Now, don't worry about the sizes. Why is the above thing 80s ribosomes? Why is the below thing 70s ribosomes? You don't have to worry about that much. Just for now, keep things as things are. And then you will see the distinction once we talk about ribosomes. So, matrix containing 70s ribosomes and circular DNA. And just like so, I'm going to use a different color for better distinction. You can see it's a double membrane structure again, surrounded by a double membrane. Surrounded by a double membrane again. surrounded by a double membrane. Projections onto matrix are known as cristae. Space between the two membranes are known as the intermembranous space, and the final point, energy is released in the form of ATP. Simple. Now, the formation of ATP is a much complex process. Fortunately or unfortunately, you don't really have this at AS levels. You will be talking about this at A2 advanced A2 levels. Your chapter 12 would be about cellular respiration and the entire process of how ATP is synthesized. For now, this much of information about the mitochondrion is more than enough, and I'm going to tell you something fun about the mitochondrion as well. Now, it's really a theory, and this theory is referred to as the endosymbiont theory. I'm going to word things out like this: endosymbiont theory. This is the endosymbiont theory. What exactly do we mean by the endosymbiont theory? Now, do you see a mitochondrion is actually a pretty large thing. It's not that large, not as large as the nucleus, but it's pretty large. That's why even in this animal cell, you can distinctively see that we have two mitochondria and they're pretty large and protruded like this. Do you not think, when you look at it, do you not think, if this guy was present outside of the cell surface membrane, it looks like a little bacterium, or it looks like a complete sole individual organism. It looks like a complete sole individual organism, right? It does. So the endosymbiont theory simply suggests, there's more to this, I'll just give you the rough surface level idea. Simply speaking, it suggests that once mitochondria were free living organisms. They were free living organisms that found their way into human cells and took residence. So now they are part of us. They are now part of us. Simply a theory. There's evidence to prove it, yes. It's a very interesting theory as well. You know about symbiotes and how interesting symbiosis is. So if you are interested, do give it a small research, and I'm pretty sure things are going to help you a lot. With that said, I will move on to the next parts. We'll be talking about the long-awaited ribosomes. Ribosomes are extremely small, only 25 nanometers in size. Only 25 nanometers in size, and like we discussed, ribosomes are the site for protein synthesis. Ribosomes are simply the site for protein synthesis. Or, in more detail, this is where mRNA is translated. Protein synthesis is a complete topic coming up, in that you'll learn much more about protein synthesis, transcription, translation, but up until this level, you do have a basic idea of protein synthesis. So simply speaking, you know that ribosomes translate mRNA, and here I'm going to give you a very important point in red color. Ribosomes do not have a membrane. Now everything we discussed had a membrane. The mitochondrion, the nucleus, the cell surface membrane itself is a membrane. So the cell also has a membrane, but ribosomes are one of those things that do not have a membrane. So we call them as being non-membrane bound, as being non-membrane bound. And ribosomes are now I didn't really draw ribosomes because it's useless to draw dots, but I'll draw dots here. So ribosomes are composed of, here's the deal, we have ribosomal RNA. Ribosomal RNA, that's too long, right? So you call it rRNA. Ribosomal RNA and proteins Now here's the deal. Now before we talked about ATS, I keep saying here's the deal too much. I think I should drop that verse. I'll try, okay? We talked about ATS, we talked about 70S. What is S? What is 80? What is 70? Here's the distinction that I'll give you, and you're going to remember this for the rest of your life because this is going to come up a lot no matter what you do. For the distinction, let me use a pretty little blue or a pink. Blue, green. I'll use a green. Okay, that's good. Ribosomes are actually classified according to their size. So we have specific size quotations. We call them S units. S units mean Swedberg units. S units in the sense Swedberg units. There's not that much detail you need to know from this. Just know that S means Swedberg units. Eukaryotic ribosomes, meaning developed ribosomes, are going to be ATS ribosomes, and they're large, and they are large. So if it's human ribosomes, it will be ATS. On the other end of that spectrum, we have prokaryotic ribosomes. Prokaryotic ribosomes are considered as being 70S. So whatever ribosomes you're going to find on the human, meaning us, a eukaryote, it's going to be ATS ribosomes. That's why we have ATS. But you know, the mitochondria, it was once suspected to be a prokaryote, a bacteria living on its own. That is why they have 70S ribosomes. This is also true for chloroplasts. We'll do that once we discuss about cellular organelles of a plant cell. There's not that much there. So 70S ATS, that is the distinction. If it's a eukaryotic ribosome, it's ATS. If it's a prokaryotic ribosome, it is a 70S ribosome. Jumping onto a similar thing, 5, 6, 7, lysosomes. Lysosomes are also not that large or that small, 0.5 micrometers in size. Here's the deal about lysosomes. They are not non-membrane bound, but they are single membrane bound. One membrane. Single membrane bound. This is the most important function of a lysosome. Lysosomes contain hydrolytic enzymes. I'll elaborate that. I'm going to get rid of enzymes. I'm going to bring it back, but contain hydrolytic digestive enzymes. That's the function of a lysosome. Contains hydrolytic digestive enzymes. That's the part. What can it do? So what can we exactly do by utilizing hydrolytic digestive enzymes? What we can do is we can break things down. So it's going to break things down. Breaks down unwanted structures. I'll just give an example. So things like old organelles, it can break down, and it can break down a lot more things, not just old organelles. It's not that hard to learn about lysosomes. They're simply the sorry, that's very bad grammar. They're simply the destroying agents of a cell. They're completely capable of destroying completely proteins into nothing. Using what? Using their hydrolytic digestive enzyme array. Now, let's talk about a very misunderstood and confused type of thing. Microtubules. Talk about microtubules. Microtubules are very simply long hollow tubes that make up the cytoskeleton of a cell shape. Now I know we draw cells like this. Okay, pretty perfect. Boom, that's it. But the reality is, to maintain the shape of this cell, there's so many microtubules. Long hollow tubules that make up the cytoskeleton, cell skeleton of this, so that the cell is capable of retaining its shape like so. Cell is capable of retaining its shape like so. Microtubule arrangement is very interesting. It's not hard, but it's very interesting. I'm gonna draw how a microtubule looks like here. Should I draw it on the left or right? I'll draw it here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yeah, something like this. Okay. Now the number is not really accurate, but here you should have 13. Number is not really accurate, meaning my drawing is not accurate. There's, I think, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. I'll just get rid of one there. I think that's 13. If it's not 13, please, you can, I don't know, you can scream at me that I don't know how to count. That's okay. I agree. I do not know how to count. I'm very bad at maths. I agree. 13, we call them protofilaments. 13 protofilaments. 13 protofilaments, and typically the size of this is about 25 nanometers, like a ribosome, very small. And each of these protofilaments are even smaller, roughly 3 nanometers in size. And these things are formed from, now these are tubules, no. These things are mainly formed from two proteins known as beta tubulin and, you might have guessed it, alpha tubulin. I should probably have mentioned the alpha first and then the beta, but never mind. Beta tubulin and alpha tubulin proteins make up the microtubules. And I will write that as a statement made up of dimers that are formed by alpha and beta tubulin. And then these dimers, so simply speaking, this is the idea. beta tubulin, and then that connects to alpha tubulin. And then those things can polymerize basically. So these dimers polymerize. Dimers polymerize to form a protofilament. To form a protofilament, meaning, so now that's a dimer, no? So multiple dimers. It's one dimer, two dimers, three, and like so it goes on. Life goes on. Like so. First we're forming a dimer made out of beta tubulin and alpha tubulin, and then those beta and alpha tubulin dimers polymerize to form a protofilament consisting of 13 units. That's the idea. We have 13 protofilaments. Now I highly encourage you go on to Google and give a small search about microtubules and really try to understand the 3D structure, because this is once again a 2D picture and I can't really give you much more of a perspective, but these are actually 3D structures. You understand that, right? These are actually 3D structures. All of this, not just this, all of these things, they're 3D structures. We're just used to seeing their 2D forms a lot, but you have to always remember biology is 3D. Biology is always found in 3 dimensions, if not 4. So it's very important that you get the proper idea into your mind that these aren't 2D structures, they are in fact 3D structures. On to 5, 6, 7, 8. No, not really 8. So wait, 5, 6, 7, 8. That's not 7, that's 8. Okay. And the final set, which would be 9. Now we're talking about, this is also a very confused thing, centrosome and centrioles. Centrosome and centrioles. Learn it exactly in the way I'm going to teach this to you. Otherwise, you're going to just end up getting confused. Now, what is the centrosome? The centrosome is not really an organelle, if you will. Now, if you go here, if you go back to our diagram, you can clearly see I have talked about centrioles, but there's not a mention of centrosome or centrosomes, right? That is because it's not an organelle of its own. The centrosome is simply a region. Okay. The centrosome is the region where two centrioles are found at right angles, to each other. It's the region, you see, and this region acts as the microtubule organizing center. Typically, we refer to them as the MTOC, microtubule organizing center, microtubule organizing centers, MTOCs. Okay, I'm gonna draw centriole. Don't worry about the structure, I'll make this clear in a bit. So what's right, so something like this, and there's a second centriole, like so. Don't worry about the structure again, I'll make that clear, and you can clearly see the 90 degree, I think I should mark that with a separate color, 90 degree difference right there. Centrosomes and centrioles, they're right next to it, like so. And typically this is not really that important, but we refer to this main one as being the mother centriole and this as the daughter centriole. It's not that important, but I think I should mention this. And that wording, oh my god, does not look a bit like daughter. That's a U, okay. Daughter centriole. Okay, now we know what a centrosome is. Now onwards to figuring out what exactly a centriole is in more detail. A centriole is simply formed. So we're talking about one unit. This is a centriole, this is a centriole. So a centriole is formed by 9 triplets of microtubules. It's formed by 9 triplets of microtubules. What on earth does that mean? Here's the thing. A triplet is 3, so I'm gonna draw 3. 1, 2, 3. Just like so, we have 9 triplets. So that's the second triplet. 2, 3, 4, 5, 6, 7. There's a 9, 1, 2, 3, 4, 5, 6, 7, 8. Let's draw another one. Let me space things a little bit more. If it does not make sense, if this diagram does not make sense, it will in a bit. Please hold on. 1, 2, 3, 4, 5, and here's the last one. Nope. Wow, my lord. 1, 2, 3, right. And then, of course, they are 3D structures. Do you see? It's a long type of structure. It's a long type of structure. So that is a triplet, and you can clearly see we have 9 triplets. That's why this structure looks weird. That's why I just didn't draw 2 cylinders. They're not 2 cylinders at all. They're complex structures formed from 9 triplets of microtubules. We have 9 triplets. There's nothing hard here. Understand what it's saying. We have 9 triplets. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplets of microtubules we have, and therefore that is what we call a centriole. Why are they so important? Let's talk a bit about their functions. Why do we now? First of all, we know why microtubules are important. That's because they are going to be taking care of the cell's cytoskeleton. Now, specifically, some other functions would be, during mitosis and meiosis, sister chromatids have to separate. No. Separation of sister chromatids during nuclear division. During nuclear division by spindle fibers. Now, do you remember these spindle fiber things that were separating sister chromatids? They are actually part of this MTOC and microtubules and the cytoskeleton of the cell. Then they were just not being produced instantly all of a sudden. They were always part of the cell. They were always part of the cell's cytoskeleton. So are centrioles and centrosomes. They organize microtubules, MTOCs, and they are responsible for the separation of sister chromatids during meiosis or mitosis by spindle fibers. Okay? And sometimes cilia and flagella and their beating movements. What does that mean? Things can have cilia and things can have flagella, right? Flagella and cilia are also made out of microtubules. Interesting, right? They are simply extended microtubules. Extended microtubules coming all the way from microtubule organizing centers, and therefore they are also part of the microtubule community. With that, we have all the animal organelles in a specific list. Learn them in the specific list. Not, you don't have to, but it's much easier if you learn it in this specific list, because it's very in a very chronological order of what and how things are happening. That's the idea. Okay, with that, all of these organelles regarding an animal cell are over.